Simon is here.
Yeah.
So you can sort of start now.
Okay. So everybody can see the slides, I presume. They're on the right part. Are they, Katrin? Just checking.
Yes, they are.
Right, cool. Okay, excellent. So thank you for the opportunity to talk about these two projects today. There's two, one which is a lab-based project and the other one which is currently underway, which is a field-based project. And both Warish, Jatinder and myself will cover some of the components that we've been into this. So just as a quick overview, what we're looking at here, in one project, we're looking at a range of microorganisms that can cause disease. And effectively what we're looking at predominantly are those that come from enteric in origin. So those are the ones that are excreted by humans or animals in their feces, get into the waterways, and then can cause a problem down the track to people. So SEQ water is required to provide a good quality water that can be used for drinking, and therefore the more that is understood about the processes in the waterways, the easier it is for SEQ water to supply a good quality water that can help protect people's health. Where we're coming from is actually having a look at the pathogens, how they behave in the environment. Now we know that they can come from that first point. Here we're just talking about a cow, but it could be sewage discharge or other animals. And then there's processes where the pathogens can move through the environment. And as they move through the environment, they are removed as well by natural processes. And what we're trying to do is gain an understanding of what happens in those processes, which allows SEQ water to better control under a range of circumstances. So we're not just interested in what happens when everything is fine and there's good sunshine, but we need to know what happens when there is long periods of drought and low flow rates, or high rain periods and high flow rates, and to understand the processes. And sometimes, as we know in Queensland, we can go from a long period of low flow rates to a sudden rapid period of high flow rates. And therefore we need to understand the processes with the pathogens to be able to work out what's the change in risk over that sudden change in processes. So that's effectively what I've been saying here, but to put it in a context, what we want to do is understand how pathogens are transported in the catchments and their survival, which allows then SEQ water and others then to work on the, to assess the public health risk. And like I said, that's not a static situation. That is a situation that can change with changes in climate conditions. That is going to be done by other people. We're providing the data that can be put into hydrodynamic models, climate models, all those other parts that can be brought in. At the moment, the amount of information that is coming from Southeast Queensland subtropical is very limited. We did do some work back in the Urban Water Security Research days, but that was still quite limited, and it was actually limited to Lake Wivenhoe. So what we want to do is more spread it out, so we've got that whole idea of the Queensland region and the subtropical. So it's broader than just the one location of Lake Wivenhoe. It will help with the interpretation when SEQ water needs to go out and do field testing, because it'll help understand where to test and where's the most appropriate place to test. It allows us to put it into quantitative microbial risk assessment and also into other models. So we can actually do health risk assessments, so it can help that decision making down the line about what needs to be done. And effectively it also helps SEQ water. So if SEQ water needs to make a decision about a process, for example, moving, not allowing something to happen in the catchments or putting a case that it can't happen, then you've got some sound scientific data which helps you justify that decision, which actually from a legal point of view and a governance point of view gives you the security that you know that your decision making is bound on sound scientific data. So the approach, very quickly before I hand over to Warish, is that, as I said, there's two related projects here. One was a laboratory project where we looked at a range of pathogens. those indicators, the FIBs, which are the fecal indicator bacteria, mostly E. coli and enterococci, and a range of microbial source tracking organisms. And the microbial source tracking organisms are those that are specific to certain animals only. And so we can say if we pick up that a specific microbial source tracker, it's come from humans or it's come from cows. And that allows us to also help work out the risk of where these pathogens may be coming through from, because some pathogens, such as Cryptosporidium, which is the major bit we're talking about, can come from a range of warm-blooded animals, not just humans, although others like viruses are from humans only, mostly. So therefore we can actually work out what the risk is by understanding those microbial source tracking. And then the longer one, as I said, which all three of us will present to, is a project that's going on at the moment focusing on Cryptosporidium in more detail, because it's a particular pathogen of concern due to its environmental stability and its resistance to various forms of disinfection, particularly chlorination. Right. That's in a nutshell why we're presenting to you today. I'm going to hand over to Warish now to talk about the first shorter investigation that was done under laboratory conditions. Over to you, Warish.
All right, so this project started in February 2019 and it ended in February 2020. So we had basically a hypothesis that microbes and pathogens decay faster in subtropical waters than colder regions due to elevated temperature and sunlight exposure. So we conducted thorough literature review, but we noticed very limited has been done on the decay of microbes and pathogens in SAQ and subtropical waters in general. So we conducted some laboratory experiments to determine the fecal indicator bacteria, associated MST marker genes, and 3 reference pathogens in 2 freshwater microcosms mimicking WAPA and Wibenolex. Move to the next one. So these are the microbial targets and methods we chose for this study. We used E. coli, which is a general fecal indicator bacteria, and for E. coli, we used culture-based methods. We used 2 marker genes, HF103 and cross-patch. These are completely specific to only sewage. For these methods, we use qPCR because these marker genes, cannot be cultured. And for 3 pathogens, we picked up Campylobacter species, which is also called Campi, adenovirus 40 and 41, and Cryptosporidium parvum, which is called Crypto. So these pathogens basically cause diarrhea, fever, and abdominal pain in infected humans. So these are the treatments and experimental setup. We basically collected, we chose 2 water types, WAPA and Wibeno. Because their characteristics were different, I think WAPA had high turbidity compared to Wibeno, and also Wibeno, I think pH was greater than the WAPA. What we did is we brought we also chose 2 temperature, 15 degrees centigrade and 25 degrees centigrade. These are average winter and summer temperature in SAQ. So what we did is that we brought the water samples in the lab and then we seeded Cryptosporidium, and also we seeded sewage into the water sample in dialysis bag. And then we basically kept the dialysis bag into this water bath set at 15 degree and 25 degrees centigrade. And then we created sunlight using an artificial light. So the light was on for 11 hours per day. And then we periodically collected sample over 28 days. So after collection of a sample, what we did is we used qPCR, culture-based methods, and microscopic methods. So we quantify all the pathogens and markers, and then we produced a decay curve using sort of a chick model. So decay is denoted by K. So I'll show you some now results that this slide is showing the results for E. coli and sewage-associated marker genes. As you can see that the results are expressed in T90. So T90 is the reduction time for a pathogens and basically expressed in days. So you can see that E. coli persisted very shorter, just 1 to 2 days. And for Wibeno dam, I think that it was only 0.3 days for 1 log reduction. HF103 and cross-patch, they persisted a little bit greater than E. coli, which is expected because these are DNA targets and E. coli is viable. So this slide is showing basically decay of Campi, adenovirus, and Cryptosporidium. Decay of Campi and adenovirus were greater at 25 degrees centigrade than 15 degrees centigrade for WAPA microcosms. However, for Wibeno microcosm, this was not the case, and I'm not clear why is this, but this could be due to the water characteristics as well. For adenovirus and Cryptosporidium, for adenovirus and Cryptosporidium DNA, for adenovirus, they persisted longer than the Campi, and but again, yeah, as you can see that 15 degrees centigrade, they persisted longer, and 25 degrees centigrade, they persisted shorter. So the Cryptosporidium, viable Cryptosporidium also persisted up to 19 days for 1 log reduction. So the interesting thing is that if you go back and if you look at the E. coli sort of a T90, which is only 1 and 2 days, and then you see the Cryptosporidium T90, which is 13 to 90 days. So clearly suggesting that your E. coli would not be a better indicator for pathogens. So these are some key findings. Culturable E. coli inactivated faster than PCR-based sewage-associated MST markers and pathogens. Water types, WAPA versus Wibeno, did not influence the decay of the tested microorganisms and their DNA. And the decay rates of most targets were greater at 25 degrees centigrade compared to 15 degrees centigrade. So the hypothesis we made in the beginning, I think, is somewhat correct. Also, this suggests that pathogens will decay faster in the summer compared to winter season. So far, we are able to publish 1 paper in Stoughton, and we are also working on another review paper and currently performing a meta-analysis to determine factors that drive decay in subtropical waters. So this is the sort of the first project, and now we'll talk about the second project, and I'll present a component. So the title of the project is Transport and Fate of Cryptosporidium in Subtropical Waterways and Storages. So this is the larger project ongoing, and it has 3 major components. First one is the decay of crypto in cow poop. Second one is reduction of crypto through transportation from cow poop to the waterways. Jyotinder has took a lead on this project, and the third one is decay in waterways, and Simon took a lead on this project. So decay in cow manure. This component of the project has been completed, and now I'll present some data. So this slide is showing the experimental protocol. We determined the decay of crypto in cow poop because cows are the main reservoir of crypto in the catchments. For example, a cow can shed up to 100,000 Cryptosporidium per day. So considering if you have 100 cows in a catchment, and if 10% are carrying crypto, that's a huge load. Through the literature review, we also find out that only 5 or 6 studies basically have determined the decay of Cryptosporidium in cow pat, and they all were done in laboratory conditions. But for our study, we determined the decay in the field conditions. For this study, we chose 2 seasons, summer and winter, under sunlight and shaded conditions. And for sunlight and shaded treatments, we placed the microcosms on the surface and subsurface of the cow pats to show the impacts of placements on the decay of crypto as well. So this was really a fun project. We had to collect some cow poop sample from Malini Dairy Farm. We brought the cow poop samples in the lab, and then we seeded Cryptosporidium parvum as the known concentration. I think it was 10 to the power 5 per 250 milligram of feces. And then what we did is we went to the field. We set up the experiments, one in summer, which was at Gatton, which was at Ipswich, and the other one in winter, which was at Gatton. So what we did is after this experiment ran for almost 60 days for the summer one, and the winter one ran for, I think, 90 days. So we again periodically collected sample from the field over 60 and 90 days. We brought the samples to the lab, we extracted DNA, and then we sort of tested with PCR, and we followed the same path as the small project. We determined the decay K value and also determined the T90 from the K value. So these are the results. So this figure is showing the experimental setup cited at switch, and you can see the decay curve. They were pretty much linear. So the result is, as you can see, that sunlight for surface and subsurface, the decay was faster compared to the shade. So this slide is showing the decay of Cryptosporidium DNA in winter. Again, the results were pretty much similar. You see the same thing that decay was faster at the sunlight compared to the shade. So key findings: Cryptosporidium DNA persisted for up to 60 days in cow fecal samples in sunlight and shaded condition in summer. Cryptosporidium DNA also persisted for 90 days in cow fecal sample in winter. The Cryptosporidium decay was significantly faster in ambient sunlight conditions than shaded conditions in both summer and winter. So currently we are taking some statistical analysis to find out correlation among temperature, sunlight, and T90 values to find out parameters that may have influenced the decay rates. I'll hand over to Jatinder now.
What, you're gonna flick the slides because I don't have any.
I'll do that.
Yeah, okay. Okay, so so I'll talk a bit more about what we did with the overland flow experiments. So we did 2 experiments. Next slide, please. One to just find out how wetting actually takes effect in a dry soil and how much runoff could be actually yielded out of that one. And this is how we proceeded. Next slide, please, Boris. Or Simon, I don't know who has the control. Okay, so smack bang in the middle, you can see what the situation was in September when we went out to find out if our experimental plan will work. The idea there was to actually demarcate 1.5 meter by 1 meter field experiment site and try to wet top 500 mm with water and then see how the water flows. And also we recorded moisture content of the soil at a depth of 10 centimeter at 6 different sites. And what we actually realized quite quickly was basically if the rainfall is about 80 mm, it took about 20 minutes to actually let the water flow across over the 1 meter area, and then some water collected, not much, at 1.5 meter. Then we actually went in the winter period, the summer period in a month later in December. Towards the end of December, then due to the rainfall, the condition at Ivanhoe actually changed, where you can see clumps of grass growing, and this is how it actually looked like. I'll go in more detail a little bit later in the next slides. Can I have next slide, please? Okay, so this is the initial setup for soil wetting experiment. So the angle of the rainfall head was actually designed in a way that it only wetted top 500 mm, and we wanted to know how long does it actually took for water to transfer from 500 mm to 1 meter and 1.5 meter. So the red stations are the soil moisture sensors, and we added actually dye in there to actually visually inspect whether when the soil is too dry, whether there will be preferential flow or how does the flow actually takes place. Next slide, please. Okay, so here is the information. So in this graph, what we can actually visualize is with the amount of rainfall, the moisture content actually goes up, and the light blue dots are basically the station 1 and station 2. These are the moisture content probes 1 and 2, which are actually sitting smack bang in the middle of 500 mm zone. So as you can actually see, the moisture content actually increased quite rapidly in that area, and there was actually lag you can actually see from station 6, which was in 1.5 meter area. But within 20 minutes, so there was reasonable saturation, and after that the water starts actually running off. Next slide, please. So next round of experiment was by seeding Cryptosporidium into cow patties. With the cow pat we collected from Malini Dairy and subdivided into two components, 300 grams each, and seeded with Cryptosporidium up to 10 to the 6 per gram. After we have done that, we deposited them pretty close to where red dot 1 and red dot 2 is in the middle to actually simulate presence of contaminated cow fecal material on the soil. And then we applied about 108 mm of rainfall, and the slope of the plot was 5.5. We regularly took samples along the flow path at these 6 stations, which I'll go into a little bit more detail. So the way we were actually collecting those samples was we installed 3 U channels to suck water into our, with suction pump into our bottles. So one was aligned at 500 mm, the other one was at 1000, and the third one was on 1.5 meter. Next slide, please. So the idea about this intermittent rainfall was to not only allow water runoff, but also some time in between for rain to actually penetrate vertically down, rather than actually creating a rainfall. So what we actually saw there was from the cow pat, there was a kind of a regular or slow release of Cryptosporidium with water. So at 500 mm distance, we regularly collected over the duration of the experiment, which was 50 minutes. Every 10 minutes or less than 10 minutes, we were sampling. So we regularly saw Cryptosporidium presence. Over time, it actually does reduce during the duration of 50, but it was a constant slow bleed. Whereas if you look onto the 100 mm side, then it took about 8 minutes or 10 minutes or thereabouts for first indication of Cryptosporidium present there. And then there was also regular sort of bleed thereon. Reminding you that we were actually applying this rainfall over 1 meter block. What that actually led to was slow release of Cryptosporidium and a complete runoff for next 500 meter. And at 1.5 meter, we did see Cryptosporidium after 20 minutes. And after that, with this amount of rainfall during the duration of 50 minutes, we saw 3 instances where the Cryptosporidium was actually present at the same level, which is about 4 logs. And after that, we did not actually see any Cryptosporidium in there. Next slide, please. During the this experimental stuff, the ground temperature was constantly above 45 degrees centigrade, and it was completely sunny. And you can say the intensity of the light quite high during the duration of the experiment. This temperature and light intensity hobo was actually placed on the ground surface next to where we were actually doing this experimental work. Next slide, please. Okay, so what we did then was we actually realized that there is this kind of a no visual disintegration of cow fecal material with the amount of rainfall we were actually applying. Then the second round was to actually kind of simulate high intensity constant rainfall event. So what we did there was we actually applied the rainfall onto the cow paddy and within the 1 meter area equivalent to 500 mm of rainfall, and then monitored because there was too much water flowing all across 3 channels. We constantly took samples at 500 mm, 100 mm, and 1500 mm, and also what was left beyond 1.5 meter collected into the sump. So as you can see in here, there is a kind of constant presence of Cryptosporidium all across the sites where we actually collected. On top of the bars, there is the volume of the water which we have collected, then actually converted the numbers to per gram of solid material in there. So what you can actually see in here is the amount of rainfall actually determine how far the Cryptosporidium go, and also what is in the cow pad is never, so this photo after the end of the experiment on the right is actually showing, even in spite of this 500 mm rainfall over 20-30 minutes period, there is still cowpat left, and there are Cryptosporidium in there, which might actually bleed if the experiment has run a bit longer. Next slide, please. So here is the end result. So the key findings are there is a slow release, as I mentioned earlier. And if we actually calculate from this experiment, which I hate to because there's only one experiment we have done in summer, we're waiting for 2 more, then we will drive those numbers. So you can expect kind of 0.84 log over a 1 meter distance if you have about 108 mm of rainfall. If the rainfall actually increases, your log removal will actually decrease as well. But the good news is with this amount of rainfall, in the ambient conditions, Cryptosporidium does not move beyond 1.5 meter, which was the main distance we covered in this experiment. High rainfall will create excessive conditions of Cryptosporidium movement from kind of water runoff volume will also determine where the Cryptosporidium will end up with. In the future experiments, we want to do 2 winter runoff experiments. And we're also trying to actually select the second site, and we will also do in-depth analysis in terms of what sort of log removal we can expect with rainfall and the slope of the land. Well, that one I'll turn to Sami.
Thanks, Jatinder. So I'm going to give the last little bit of what we're going to present today, which is if viable Cryptosporidium ends up in the waterways, what happens to them from there? As Warris showed in some of the small in-lab experiment, there is a period of decay that he could monitor. So we were looking at that again out in the real world, and we were seeing if there was any change and looking across a broader sites in southeast Queensland. There are other, and I'll talk about what we did in that. There are 2 smaller experiments we did, which just because of time we're not covering today, which is the absorption of Cryptosporidium oocysts to soil materials and its subsequent sedimentation, either absorbed to materials or not. But we have covered that. If anybody wants to know any of that information later on, we can provide some information, anybody really interested in that. So we've got a series of experiments that we're running in the waterways in southeast Queensland. We've got a series of parameters that we're looking at. There's the seasonal impact, similar to replicating what Warris ran in the laboratory, but out in the field. So that's winter and summer, effectively. And just for the sake of simple, we're calling that temperature experiments. In the literature, we know that dissolved oxygen may have an effect on Cryptosporidium decay. So we've had a look at whether the oxygen is present or not. And certainly some of the dams are deep enough or have conditions suitable that you will have different layers. So Wivenhoe and Whopper Dam, for example, if you get down to the base of the reservoir, there is very little to no oxygen. If you're up at the surface, you have basically full saturated conditions for oxygen. So we're testing that out. There's a series of biological activity experiments because we know that certain microorganisms can influence the survival or the decay rates of pathogens when they get into the environment. So that's what we call biological activity. It's predation, it's production of antimicrobial compounds, those sorts of things. And then the final one is sunlight. So if you've got Cryptosporidium towards the surface of the reservoirs and the rivers, what effect does sunlight have? You can see we've done them in Wivenhoe. We have a site at the Mid-Brisbane River, and Whopper Dam are the experiments that have occurred. And you can see there's a number of them have occurred both in summer and winter since the start of the experiment so far. They're very simple, predominantly. Apart from the sunlight experiments, what we do is we collect water from the reservoir or the location. We have what's called chambers, and you can see a schematic, a split diagram of them here. You bring them together. So we have a volume of water, usually about 1 litre, which we seed with Cryptosporidium, and then we inject that water into a number of these chambers, so, enough chambers so we can come back every sample event and collect 3 of these chambers and collect the water out of those chambers and analysed the number of viable Cryptosporidium left in that chamber at that time. And from that, we can get a decay curve. Because we've added the Cryptosporidium to the water and given it a really good mix, and then added to the range of chambers, and those chambers are collected randomly, it takes out any statistical variation. The other thing we can do, such for the biological experiment, is we can actually remove the microorganisms out of that water before seeding with the Cryptosporidium in one set of chambers, and then have another set which still have the natural microorganisms present in the water. And so we can compare, that's how we assess the effect of the biological activity. Or for the dissolved oxygen, we have a series of chambers at the surface, and then we have a series of chambers down at the depth, basically sitting on the bottom of the area that we're testing it in. And just on the right here, we actually do it by look at the viability by microscopy methods using a vital stain. And so if they stain blue and only blue, then they're viable. If they stain blue and red by the different dyes, they're non-viable. And you can see here, for example, here's a viable Cryptosporidium oocyst, and here's two non-viable Cryptosporidium oocysts. And we can monitor the change over time. So I'll show you what the results are from here. If we look at seasonal decay, and this is just one example, the decay curve, again, similar to what Warwick saw in the laboratory, it's pretty much linear, at least in Wivenhoe Dam here. You can see this is the Wivenhoe setup that we use. Originally, we were trying to position it against the profile that's here, just so we had a position to put it. Unfortunately, we were competing with the profiler with the local cormorants who are nesting on the site, so we actually ended up moving a little bit away from the profiler to do our experiments. But you can see, this is the mid-Brisbane, I should say, in summer, and it's we've got a decay, but what we actually found was that towards the end of the decays, we could see a little bit of slowdown. So we would actually go to a quadratic decay rate and having a T1 log reduction, which is the equivalent of a T90. But because we're getting a biphasic decay, you can see it starts off quite fast in winter in Lake Wivenhoe, and then will slow down. And typical what we see in a lot of ecological studies, you have a proportion of the population of those oocysts that are a bit more resistant and will persist for a longer period of time. So we lose a large proportion of the Cryptosporidium in the early stages, up to about 60 days, and then the last little bit last a little bit longer. And certainly in winter conditions, you see faster than we're seeing in the mid-Brisbane. Certainly because of the way we're setting up the experiments, all the dissolved oxygen and the biological at the surface are also going to be used as part of these seasons. So I'm only giving you the ones which are done dedicated to look at seasonal impacts, but when we come back to do the full analysis down the track, we'll have a lot more data for the impact of seasons. When we look at dissolved oxygen, Lake Wivenhoe, we certainly don't see too much of a difference at the surface versus the depth, which is going for that. But when we get to Whopper Dam, which, as Warwick pointed out, is a very different dam, it's got a much higher organic load, we see a much more increased biological activity. You can see that there's a difference between the decay at the surface, which is quite biphasic, and a decay at the depth, which is quite linear and only one decay rate being obtained. Which shows it, and we see that there are pictures, and when the final report comes out, there's differences that we see at Whopper Dam for the removal that's happening. So we'll be actually doing another experiment in Whopper Dam, which I'll talk about in the end, which will study the impact of the microorganisms. That's going to be done next summer. The influence of biological activity, like I said, this is actually looking at the predation and antimicrobial activity of the natural flora, and this is something we've seen in a wide range of other environmental samples. When you get pathogens that have come out of intestines of an infected animal, be it humans or cows, we can actually then, when they get into the water, they're quite susceptible to the impact of the natural microorganisms, which are more established and able to outcompete and and in some cases predate directly on the pathogens. And again, you can see that the mid-Brisbane River, which is actually done in winter, is actually quite, when we remove the microorganisms, there's a significant difference between what we see with the microorganisms present at 41 days for a 1 log reduction versus 274 days. So the microorganisms in the mid-Brisbane are really driving decay in that conditions. It's a very shallow system with a standard temperature across. If we go back out to Lake Wivenhoe, we do see a difference, but that difference is actually much smaller and statistically is becoming almost not statistically significant. Again, the next stage of the project is to actually start to compare across these different sites, and we'll be doing another biological experiment out in Whopper Dam to see what the influence in a highly nutrient, very active reservoir lake, what the difference we'll see from that. And the last one, just to show off, is sunlight. And this is the only one that's different. We're not using the chambers in this situation because the chambers are made of Teflon, so light doesn't get through Teflon. So it's a bit hard to see, but you can see in our floating little rig here, there's little vials. These are scintillation vials, which are low potassium borosilicate, which we know allows a large proportion of UV light through. We've actually tested that out in the laboratory. And what we have is we have a series of the vials sitting here that are exposed. They sit just below the surface. And then we have a series of vials onto this side, which are set up, which are surrounded by aluminium foil. So they're not exposed, but kept in the same conditions. And what we do is we just have them, this is at Lake Wivenhoe again, they're out, we take it out to this buoy, which is about 30 metres off the shore. We sit there for an hour, we bring it back in, collect a series of vials, reel it back out. It's an exciting day, I can tell you. And then they go back and we let them sit in the fridge for about 5 weeks, and then we stain them so that the UV has a chance to take effect. This is the only one that's available at the moment. I'm in the process of running the summer one. This is done in winter. And as you can see, there's no decay. We're not seeing any influence of this of the sunlight at less than 10 centimetres depth of water under the winter sun conditions. So we'll wait and see whether summer, which is warmer and has a more intense sunlight, actually has a greater influence at that time. As you can see, the sunlight gets up. It was a bit of a day, one of those days where it was had a few cloud covers come over, which is why you see it drop down and then come back up again. But more results have to be done with that, but just showing you one of the results available to date. This is a lot of work. We're still measuring this. What we're able to show, though, is that we can actually measure the decay rate, and we can show that it's occurring. Just natural decay by the microorganisms over summer and winter. There is a little bit of seasonal impact, but it's not a massive impact on great periods of times. We need to do a lot more on site variability, so I don't want to say whether what the impact of the site is and how great that difference is. That will come out as we do more at Hinds Dam or Alltown, our lake, and do another experiment out at Whopper Dam. So there is some impact of site variability, but whether that's big enough to make a major difference remains to be seen. And then what we're going to be doing as we start to get all this results is actually do a really in-depth statistical analysis, looking across the different sites and the different conditions that we've tested. And we've got water conditions, so we've got pH, electrical conductivity, and a range of other parameters that have been supplied. And just to see if we can see any differences across the different sites based on water conditions, the environmental conditions, and the decay rates that are going on. We really need to go in and have a look at that biphasic decay and see what's driving that. And then finally, we've got three more experiments to do, another sunlight experiment and 2 at Whopper Dam and Lake Danstown, I should say, not Alltown, sorry. Once we've got that, then what we'll be doing is taking all the results of all the different experiments from the decay in the cowpats, the overland flow, removal in the waterways, and start to construct a conceptual model of how Cryptosporidium moves and behaves in the environment out in southeast Queensland, and that then can be used, that model can then be used to work out as conditions change, whether we see climate change, whether we see different weather events, and that conceptual model can then be married to any hydrodynamic models, 3D models, or whatever else that SEQ Water is doing to help SEQ Water make better decision-making processes, particularly based around Cryptosporidium, which is such a problematic pathogen in the environment. And I've gone for long enough, so we'll finish up there and catch and see if there's any questions.
Yeah. Thanks to all three of you for presenting. That's great. We have about 50 minutes left for questions, but didn't receive any in the chat yet, as far as I can tell. So if anyone, I see that Barry has his hand up. Just talk, Barry.
Oh, thanks very much, and great presentation. I just had a question when it came to your baseline data to do with the cow poops. You know, I see that you're getting that, or you're sourcing that material from Maleny. Have you done any other research on the variability of based on their diet from around the catchments? And also, did you compare that with any sort of native scats that you might have found, that sort of thing?
I don't think we haven't, right?
No,
we haven't.
Not yet. Obviously, it's something we do need to think about, whether there's a great variation in cow manure and their diet. And certainly we use cows because of the number of cows that are out there as our model organism. We certainly could probably go back and at a later date, not this project, just because the amount of work that's involved in it, consider maybe in the final report we can actually find some information about kangaroos and pigs and goats, which we know are all out there. Their fecal load will be smaller than cows, which is why we've stuck with cows at the time. But certainly it's a good question, and we should incorporate that as much, at least from the literature, as we can.
I think there was a study from US. Basically, they shown that your diet influences bacteroides in cow feces, but we're not sure about Cryptosporidium. Yeah.
Okay, yeah, no, it was just one of my thoughts because it's quite a rich diet up there at Maleny Dairy as compared to the roughage that they'd be getting around, you know, the back end of Wivenhoe, and they'd have different microbes in their gut too, which might affect the Cryptosporidium as well. So that was just my thoughts. Thanks, eh?
I might actually add on this one. What we have actually seen, like you mentioned, that the grass, these cattle are basically heavily fed on lean grass, and the composition of the fecal pads are quite soft and fluffy, so to speak. But it could be, the diet could be one of the variables in terms of also how far the travel, not only what is coming out of the so it is definitely worth exploring further.
Yeah, thank you.
Thanks, Barry. Any other questions? Then we just received one in the chat from Ian. What is the cost impact of these pathogens on the cost of water? How does this compare? Revenue from grazing? What is the effect of distance from source? Can this info dictate appropriate buffers?
Yeah, well, that's part of what we're trying to determine. So that's where I was saying SEQ Water can make scientifically valid decisions about the level of risk. So hypothetically, and hypothetically only, you might say that SEQ Water may say that use Lake Wivenhoe just as the example, ignoring the fact that the water then travels through the mid-Brisbane, but because I'm working on a hypothetical, that based on the data, you may restrict cows having access at any distance towards the Lake Wivenhoe for 5 metres, and they have to be fenced back further than 5 metres, access to water. But once you go 2 kilometres up Lake Wivenhoe, then that becomes not a concern, because we know that the travel times and the decay rates mean that any cryptosporidium that could enter the water would be removed before they actually were able to move the distance to the dam wall where the water offtake was occurring. That's a hypothetical, just as an example. So that gives the decision-making process, because we know from anything to do with water, if you can control at the source, it keeps your risk and your costs down when you're down at the treatment end. So it means it's much more controlled, because you know that there's a consistency of water coming in, so that your control and your treatment costs can be managed at the treatment end, because the source is controlled. And that's a common thing for all waters anywhere in the world, and that's what we're trying to provide information to help SEQ Water with that process.
Great, thank you, Simon. There's one coming in from Daniel. Did these studies confirm an crypto-infected percentage of cattle in the test dairy cattle? And sorry, my window is just blocking the next sentence. 1 in 10 was mentioned previously.
Yeah, we didn't look into.
We did not, but there are other studies from SEQ Water. They looked at the prevalence of Cryptosporidium in animal fecal sample and wastewater. So that was done a couple of years ago, I think.
Yeah, we didn't do that ourselves.
Did not do that.
But we can also say that because we're adding significant.
Actually, we have some data that we did in a previous study. We find out that 2% prevalence in cow pat.
Yeah.
So, yeah.
But even if a cow pat we collected had Cryptosporidium in it, we're adding much more.
No, Simon, because what we did is we collected Cryptosporidium, but we checked it.
Yes.
Didn't have any Cryptosporidium before the
But
experiment.
I mean, what I'm saying is, even if there was a trace there, it would be. We have the numbers that would statistically not mean that trace in the cow manure was not going to make a difference to it.
Paul, you were going to add something?
Yeah, hi, it's Paul Fisher. I was just going to add a couple of comments to Barry and Dan's, I guess, questions. So Barry, we have done some past projects, not looking at, I guess, the diet of cattle and I guess the prevalence of Cryptosporidium, but at least comparisons of some wildlife and cattle. So I can send that information to you if you're interested. And Daniel, just looking at your question there, and also as Waresh mentioned, we have done studies in the past looking at prevalence of Cryptosporidium, so I'm happy to send you those papers, Dan, as well.
Great. Yeah, I think that's where Michael's comment is heading towards too. Yeah.
I probably just had one, I guess, comment, which was, thanks guys for the presentation, and I guess there's a lot of work that's gone into the project so far, and obviously there's more to be done. But it's, I guess, maybe people that aren't familiar with the topic, I guess some of the ideas seem, I guess, logical, where increased temperature might result in, I guess, faster decay, but there's a lot of work, and you guys have presented all the work you've done in just really trying to understand what those values are. So it's not just whether it's faster or slower, but just really trying to understand for this region what that decay rate is, and how things like site, temperature, low DO, and predation affect the number, which really, I think this is the only project that I've seen that's been so comprehensive at looking at on the land, in the cow feces, and also in the water at the same time. So,
yes.
We have 5 minutes left, so time for another 1 or 2 questions, if there are any further. If there aren't any other questions.
I was just going to add, Katrin, that effectively I think what we'd be happy to do is at the end of the project, as people can see, there's a lot of data here and there's a lot to absorb. But I think if we can come back at the end of the project and present it in that form of the conceptual model, with people understanding that there's a lot of significant data at the back, I think that'll also help people understand what it will mean to SEQ Water and how it can be applied.
Yeah, that's right. Yeah. Yeah, we will have a seminar at the end of the project again. Yeah.
Yes.
Yeah. If there are no further questions, then I'm happy to encourage everyone to send any through. If you can think of any questions later, and I'm happy to get back to you on them. And I will circulate the recording and also the publications that have been mentioned. And I'm going to share the slides as well. All right, so if there are no further questions, then thanks a lot again to the three of you. That was great. Thanks for taking the time and presenting to us. Really appreciate it.
Thank you for the opportunity to show the results so far.
Great.
Thank you.
Thanks.
All right.
Okay.
See you
Bye.
guys.
Bye.